Hi, this is Andriy from Med School EU, and we're going to begin our course with Section 2 of the IMAT, which is Biology. And we're going to start right from the top of the IMAT Specifications with the unit of the chemistry of living things, which is simply biochemistry. And the first topic that we'll cover in this video will be the biological importance of weak interactions. All right, so before we dive into weak interactions, we must first brush up on a little bit on basic chemistry and atomic structure. So here we have two atoms, sodium and chlorine. And first we must know how atoms exchange electrons in order to form interactions and form bonds within each other, which is just a theoretical basis of weak non-covalent interactions that we'll talk about in this topic. Everybody should be already familiar with the structure of the atom. There are electrons on this atom, and you should know that the most outer shell is called the valence shell, which contains electrons on it that are called valence electrons. Now, the octet rule here states that atoms have the tendency to prefer to have 8 valence electrons in its most outer shell, which means that atoms are stable when they have 8 valence electrons. And typically when they're stable, they're unreactive. They do not react very well. However, these atoms here, they don't fulfill the octet rule. They don't have 8 valence electrons because sodium only has this 1 electron here, and chlorine has 7 electrons in its valence shell. So both of them do not fulfill the octet rule. Now, if two atoms do not fulfill the octet rule, they're typically unstable. And if they are unstable, they're looking to become stable, and therefore they're open to be reactive. All right, so what happens when two unstable atoms come in contact with each other is that they form interactions. So typically when a sodium and a chlorine atom come together, the sodium electron here has the tendency to be given up and transferred over to the chlorine. Now, why is that? Well, because when sodium gives up its valence electron, it actually forms the second shell as the valence shell instead of its most outer shell. So it drops down to the second shell being the valence shell, and this one has 8 electrons, which fulfills the octet rule. Now, why does chlorine want to gain this 1 electron? Well, because it has 7 electrons in its most outer shell, and by adding 1 more, it becomes stable, fulfilling the octet rule. And this is the typical basis of how two atoms become stable and form interactions. And this is the basis of how weak non-covalent interactions occur. So now let's dive into our specific topic on the IMAT, which is weak interactions in living organisms. Now, atoms and biomolecules are typically held together by covalent bonds, which are strong interactions that involve sharing of electrons. Now, non-covalent interactions are weak interactions. So basically, another name for weak interactions that are specified on the IMAT, they're also known as non-covalent interactions. And if these non-covalent interactions occur in large numbers, they create a cumulative effect because on their own they're very weak. If you have just one non-covalent interaction, the attraction between the two molecules or the two atoms will be weak and it's easily broken. However, when they occur in large numbers, they form a cumulative effect and therefore they become very powerful. So let's take a look at the 4 types of weak interactions that typically occur in organisms. And here are the 4 types. Number 1, ionic interactions. 2, hydrogen bonds. van der Waals interactions, and the last one we'll look at is hydrophobic interactions. All right, so let's begin first with ionic interactions as the first weak interaction that we're going to take a look at. Now, ionic interactions are electrostatic interactions that occur between charged particles. Now, what do we mean by that? Well, it's basically two charged atoms or molecules that come in contact with each other and they form interactions that are electrostatic, meaning there's charges involved. So the typical example we have already looked at is the sodium and chlorine, where the two atoms would come in close proximity to each other. And what occurs is that the sodium electron is being transferred over to the chlorine shell. And then they both become stable. However, these two atoms, they're charged particles. They're charged atoms. The sodium has the tendency to lose this electron, which means it has a positive charge. And the chlorine has a tendency to gain an electron in order to fulfill the octet rule, and it has a negative charge. And obviously, everybody should know the universal rule that opposite charges, they attract, and same charges repel. And this is just a universal principle with chemical bonds. Another thing to know about ionic interactions is that they're strongest in a vacuum medium. However, ionic interactions are typically weaker in aqueous environments, so when they're surrounded in water, because water tends to separate the two charges that are in close proximity. So water comes between these charges and making them separate further apart, because water typically also interacts with the positive and the negative charges as well, which makes them less attracted to each other. And if we take a look at where these ionic interactions occur in bodies, in human bodies and other organisms, most prominently they occur in, as an example states here, is the amino acids. So there's two amino acids here, lysine and glutamic or glutamate, glutamic acid or glutamate. And these two are amino acids, and some amino acids have charges on them. So lysine, as you can see, has a positive charge on this nitrogen end, and glutamic acid has a negative charge on the carboxyl end. And what happens is, when these two charges come in close proximity, when these two amino acids come in close proximity with each other, they form electrostatic interactions that are called ionic interactions. So this is an ionic interaction between two charged particles. Next, we'll discuss hydrogen bonds, which is another weak interaction that occurs in organic systems. And hydrogen bonds occur when hydrogen is covalently bound to an electronegative atom. And I'll explain what this means, but first let's take a look at what electronegative atom is. Well, typically we learned in school that electronegative atom that form hydrogen bonds are FON. So fluorine, oxygen, nitrogen. However, in biological systems, typically you would see oxygen, nitrogen, and sulfur more often than FON. However, the baseline is that electronegative atoms are atoms that are very attracted to electrons. They tend to want to gain an extra electron, as we saw with the chlorine atom. So let's look at an example, what occurs and how these hydrogen bonds happen. Well, basically we have two covalently bound hydrogens molecules, hydrogen atoms bonded to a molecule with an electronegative atom. So there's two of these, and when these two molecules come together, they tend to form hydrogen bonds. Now, how does this happen? Well, it occurs through this notion of dipoles, actually. So, because oxygen is more electronegative, it tends to hoard hydrogen's electrons and it tends to take it away. So it forms a partially negative charge. Now, same thing happens with this oxygen here, and the hydrogen would form a partially positive charge. And as you can see, what happens with these partial charges occurring is that the negative partial charge and the positive partial charge would attract each other and form something called a hydrogen bond. Now, where do these hydrogen bonds occur in biological systems? Well, most prominently and most consistently, we see them in DNA base pairing. So a DNA double helix molecule would base pair. There are 4 ATCG. So there are 4 nitrogenous bases. Adenine would base pair with thymine, and cytosine would base pair with guanine. And what happens? How do they interact? Well, they form hydrogen bonds. As you can see here, thymine and adenine would form 2 hydrogen bonds together. And this is how they interact and how a DNA double helix remains a double helix. It's very difficult to disrupt a DNA structure because there's so many of these hydrogen bonds that form a cumulative effect that we have talked about previously. Now let's talk about the third weak interaction, and that would be the van der Waals interactions. So van der Waals interactions occur between 2 uncharged atoms that approach each other. And typically their attraction between the 2 uncharged atoms is due to permanent, transient, or induced dipoles. And I'm going to explain what that is with an example. However, before we get to that, I first wanted to explain that the 2 uncharged atoms only interact and form these dipoles at a specific distance between each other. So if we have 2 molecules here, they must be at a specific distance between each other in order to form these van der Waals interactions. And this distance is called the van der Waals radius. Now, if the 2 particles are closer than the van der Waals radius, they will tend to repel each other and they will not be attracted. And then if they are obviously too far apart, then they're not approaching each other and they won't form these dipoles. So let's take a look at examples of the 3 most common types of van der Waals interactions that are called that are based on dipoles. First, we're going to take a look at dipole-dipole interactions. Now, how this one occurs is that the 2 molecules of carbon double bonded to an oxygen, the 2 molecules approach each other at the van der Waals radius, and what happens is because these 2 uncharged molecules are in this proximity, they tend to form partial charges. So let's label partial charges to these atoms. Now we know that oxygen is more electronegative than carbon. It is all based on the periodic table and the position of the atoms. So oxygen here is more electronegative. Same thing with this oxygen, more electronegative. So we're going to put a negative charge here. Whoops. Negative charge and another negative charge. And what occurs with the carbons, obviously, is they become partially positive, and we put a positive charge. And as you can see, this forms a dipole that's going in this direction because of the negative and the positive that interact with each other. And this van der Waals interaction is called dipole-dipole interaction. The next example we're going to take a look at is very similar. However, this one's called dipole-induced dipole interaction. And this one is a little bit different in terms of how the dipoles are formed. Now, again, the same thing happens as these molecules. The oxygen atom is more electronegative, so therefore it has a negative partial charge. Now, what happens with let's label this carbon, it's positive. What happens with these molecules here is that because of this partial charge on the oxygen and this molecule approaching the other molecule at the van der Waals radius, what actually occurs is that it induces a dipole on this molecule. Now, typically, carbon and hydrogen do not form dipoles on their own. They do not have charges on their own. Even partial charges are not very strong on their own. Now, because of this other molecule that's beside it, it is inducing a dipole in this molecule so that they attract and they interact with each other. So what we have here is the hydrogens begin to give up its electron to the carbon in order to interact with the negative charge of the oxygen here, with this dipole on the other side. And of course, the carbon then has a partially negative charge, and this again forms the same type of dipole as in the previous example. Now, if we take a look at the last example here, we have these two molecules that are the same as we saw earlier with the three hydrogens. And how they form dipoles is quite interesting. So what they have is these two molecules come together at the van der Waals radius, and typically, again, both of these molecules are uncharged and they don't even form partial charges on their own. However, when these two molecules come in contact at the van der Waals radius, they form partial charges. And because otherwise they would repel each other, but instead what happens is when these two collide or come close in contact at the van der Waals radius, one of these molecules forms a partial charge. So we'll pick the right side molecule. We'll have its hydrogens give up electrons to the carbon, which would form a partially positive charge on the hydrogen side, and therefore this carbon would have a partially negative charge. Now, what happens with the hydrogens on this side is, of course, because of this induced dipole, you have another induced dipole on this side, and the hydrogens will start to pull electrons from the carbon, forming a negative charge. And the carbon obviously has given up some of its electrons, and it forms a partial positive charge. And this is how van der Waals interactions occur through induced dipoles. The last type of weak interaction we're going to discuss is called hydrophobic interactions. And hydrophobic interactions typically form between nonpolar molecules in aqueous environments, so in water. And how hydrophobic interactions occur is best described with an illustration. So what we have here is these three nonpolar molecules that are thrown into an aqueous environment. And as you can see, what occurs here is that the three nonpolar molecules, they tend to stick together in order to push the water out and to have the smallest surface area interacting with the water. So this interaction results from the exclusion of water molecules, and it works best because it minimizes the interaction of the nonpolar molecules with water molecules. And we're going to see this occur a lot in the formation of 3D tertiary, so 3' protein structure, where there's plenty of hydrophobic interactions occurring because proteins must form a specific structure or shape in order to function. And these formations largely occur due to hydrophobic interactions to exclude water from interacting inside the protein while it's forming its 3D shape. So in the next video, we're going to discuss the organic molecules in organisms and their respective functions.